All right, guys. So today I'm going to just quickly, before you start your video, if you're still a little bit unsure about meiosis, do a quick side-by-side comparison of meiosis 1 and meiosis 2. Both go through prophase. So remember, prophase is where you have a nucleus that breaks down. And the big thing here, the big difference that I need to see between prophase 1 and prophase 2 when you do this is that reminder, prophase 1, let's say this organism has 4 chromosomes, a human would have 46. Prophase 1 is a diploid cell. In meiosis 2, prophase 1 would be a haploid cell. All right, you should see, and one thing we will be looking for is X-shaped chromosomes. Remember, these are all X-shaped chromosomes. X-shaped means that you have the 2 copies are still attached with a centromere. Each of those copies is called a sister chromatid. In meiosis 1, just a quick reminder, we have really important are the homologous chromosomes. Those are the chromosomes that have the same information on them. So for today, we're going to pretend that there are 2 sets of chromosomes, chromosome 1, chromosome 2. The next phase, remember PMAT, is metaphase. Our key word when we went over this the first time is we thought of this as in the middle. So here, instead of lining up in single file, they line up with their homologous pair. This would be chromosome 1 and chromosome 1 are side by side in the middle. Chromosome 2 and chromosome 2 are side by side in the middle. And of course, we have our spindle fibers that are going to pull these apart. Now, big thing in metaphase 2 is that they are lined up in the middle in single file, not with a pair, because their pairs are in separate cells. Remember, this is chromosome 1, chromosome 1, chromosome 2, chromosome 2. The end result, we don't want 2 copies of chromosome 1 in each cell. We only want 1. So that's why they have to get pulled apart in anaphase. So anaphase 1 and anaphase 2, we're starting to get, they are pulled apart. So if you remember, middle apart 2, middle apart 2. So here, we did not pull apart our sister chromatids as we do in anaphase 2. The homologous pair is pulled apart. Anaphase 2, on the other hand, sister chromatid. So let's just make a note here. Sister chromatids are the ones pulled apart. Anaphase 1 are homologous pairs. Put it right here. And that's why we started this with these words. They're very important to understanding this. And finally, just like in mitosis, we have telophase 1, telophase 2. So we go prophase 1, metaphase 1, anaphase 1, telophase 2. And remember, 1 and 2. We say 2. They break apart into 2. So here we have it. These at the end are finally, we have made it to the point we have haploid cells. So we take these 2 haploid cells and they are going to start, just to kind of put this together, in prophase 2. Remember, prophase 2, very, very similar. We're haploid. Metaphase 2, we're in the middle. Anaphase 2, those sister chromatids are pulled apart. Telophase 2, last but not least, each of these cells breaks into 2. And the end result, very important. I need to see at the very end, you need to know that we are going to have 4 haploid cells. They are not the same. So for practical purposes here, these 2 are going to end up looking the same. These 2 are going to end up looking the same. But there are things that happen in, particularly in prophase 1, if you were paying attention, that these chromosomes kind of get mixed up. They swap information. So actually at the very end of meiosis, these 4 cells are completely different, which accounts for why siblings are not identical. All right, guys, so when you are done today, make sure you attach photographs or videos. All you have to do is really create each of these phases using materials in your home. So you do not have to create a stop motion video. You can just take still pictures, which would be much quicker. Keep a track of time. If you're spending more than 40 minutes on this, send me an email. Monday's assignment will be kind of a little bit lighter, so you can finish it up then. All right, guys, have a great time.